All right, so we're here in Isle Royale National Park with Dr. Alex Egan, who, among other things, is responsible for the long-term bird monitoring research here on Isle Royale, and he's going to talk a little bit about some invertebrate sampling today. So go ahead, take it away, Alex.
Yeah, one of my main jobs is to study water quality, which is a core indicator of ecosystem health. And so we study basic things like conductivity, pH, dissolved oxygen. And so one of my study sites here at Lake Ritchie at Isle Royale, we get a lot of good chemistry data. But we also like to tie that chemistry, which is influenced by atmosphere, landscape effects, and what's going on in the lake. We like to tie it back to the things that live in the water, and so some people would tie it to fish. I tie it to aquatic insects, and what I study is called a group of flies called chironomidae, and they're mostly aquatic, and they'll live in the bottom of the lake. When they emerge as adults, they'll float to the surface and split their thorax open, and the adult flies away and their pupal case remains. And so what I'm doing is I'm using just simple equipment, a sieve and a pan, to scoop up those pupal cases, and then I can identify those to species, and we can get a good sense of both abundance and diversity of chironomids that are in this lake. And one of the interesting things about chironomids is they're so diverse that they can really help to tell you a little bit about both the chemistry, so we tie it back to the chemistry, and just the general ecosystem health.
That's awesome. So how many species of chironomid flies or midges would you find here?
In Lake Ritchie, my guess is there could be 100 species in this lake.
Wow.
Just a rough estimate.
And just this was new to me too, and I presumably new to the students. So you're not actually collecting the live insects, you're actually collecting the pupal cases that were left over.
Yep. And there's a couple of reasons for that. One is the adults, once they start to fly, you're not necessarily sure, unless you use a special emergence trap, you don't know necessarily where they emerged from. And the larvae, if you collect the larvae, you can do a grab sample of the bottom material. Those are hard to identify to species. You can get them to genus, but not species. The pupal case, I think, is a perfect medium where you can get most of them to species, and they're floating on the surface of the water, so you're pretty sure what habitat they emerged from. We don't know the microhabitats, whether it was sandy bottoms or mucky bottoms or somewhere, but we know what lake they came from anyways.
Very cool. And so how do you go about collecting the pupal cases, these midges?
Well, so the dominant wind is from west to east here, so I'm in the eastern bay. There's not too much material here right now. If there were a wind or a slight breeze, it'd probably be blowing more stuff in. So I'm just looking at this surface for just stuff floating around, and if I see anything floating, there's probably midges there. And so I'll literally just dip this pan in and just spend 10 minutes scooping water through this sieve, and then filter this into a collection jar and bring it back to my lab. I'll do a subsample of about 300. Depending on the lake and the sample time, I can get anywhere from, I don't know, a couple hundred specimens to tens of thousands. And so I really need to do a subsample because you can't process tens of thousands. And then I'll slide mount them, and then under a nice microscope, I will identify them to species or as close as I can.
And then one final question about the diversity, because like 100 species sounds insane, particularly from a wildlife perspective, like very hyper diverse. Is this unique to Isle Royale, or if you go to the mainland in like Ontario or Minnesota or Michigan, would you find equal kind of diversity of these midges?
Yeah, for sure.
Wow.
Yep.
Very
That's
cool.
Pretty typical worldwide. High diversity.
Awesome. Well, thanks so much, Alex.
Yeah, you're welcome.